We've just discussed the structures and functions of the epidermis. So now let's turn our attention to the dermis, which is the deepest and generally the thickest of the two layers that belongs to the skin. So the dermis consists of only 2 layers, whereas the epidermis we saw anywhere between 4 and 5 layers, depending on the region of the body that epidermis was located in. So the epidermis has 2 layers. It has the superficial papillary layer and then the deeper reticular layer. These 2 layers, kind of unlike the epidermal layers, these 2 layers, papillary and reticular, really have no distinct boundary between them. We see that the dermis is a strong but flexible connective tissue, so we've got a tissue type change between the epidermis and the dermis. Recall that epidermis was an epithelial tissue, but the deeper dermis is a connective tissue. So recall that connective tissues have really kind of very few scattered cells within them and are primarily composed of extracellular matrix and protein fibers. But that's not to say that the dermis is completely lacking in cells, and it turns out that the dermis has a couple of very important cell types, and these cell types are listed here. So the cells that are present in the dermis are things like fibroblasts. Fibroblasts are the cells that function to build the fibers that are within the connective tissue. We have macrophages, which function to engulf and destroy debris, foreign microorganisms, other pathogens that happen to get through the epidermis and down to the dermis. So we have macrophages that patrol the dermis. And we also have a cell type called a mast cell. And mast cells are typically associated with blood vessels within the dermis. Now, when mast cells detect microorganisms, they initiate a local inflammatory response by releasing a substance called histamine. And what histamine does is that it increases blood flow to a particular area and dilates the blood vessels there. And this makes them leaky. What this does is it enhances essentially the permeability of the blood vessels, and it allows immune system cells like macrophages to leave the bloodstream, because normally they're within the bloodstream, but it allows them to leave the bloodstream and enter the surrounding tissue to fight any potential unwanted pathogens, bacteria, fungi, things like that are in that area. And so that's what these local inflammatory responses are doing, is it's promoting blood flow to a particular area, but then also enhancing capillary permeability so that white blood cells can leave the blood and enter the surrounding tissue to fight pathogens. Mast cells also secrete another chemical substance that's called heparin, and heparin is an anticoagulant, and that prevents blood clotting. So this is just another substance that enhances blood flow to that particular area. The dermis, being a connective tissue, is vascularized. So unlike the epidermis, which we saw has no vascular tissue within it, so the blood vessels that are in the dermis not only nourish the dermis, but also nourish the deeper layers of the epidermis as well. So we see the vascularization is important for both the dermis and the epidermis by close association with the dermis. And it is also very well innervated with both nervous tissues and sensory receptors. So this image is a section through the dermis, and it really just kind of gives you an idea of the rich variety of structures that are within the dermis. So we see nervous system tissue, we see the coiled ducts of sweat glands, we see a number of blood vessels scattered through dermal tissue, and we even see some adipose tissue as well. So there's a good variety of other structures that we see in the dermis that we just did not encounter in the epidermis. So let's begin at the superficial layer of the dermis, and this is the papillary layer of the dermis. This layer sits directly up against the stratum basale of our epidermis. So the papillary dermis is what makes contact with the epidermis. It is composed of areolar connective tissue. Recall that areolar connective tissue is very loose connective tissue, but it's also very well vascularized, so it's helping to bring blood vessels in close proximity to the epidermis so that the deeper layers of the epidermis can be nourished by it. Areolar connective tissue also has a number of immune system cells that are associated with it. So any microorganisms or any foreign pathogens, dirt, debris, things like that, that get through the epidermis can be dealt with in the papillary layer of the dermis due to the high number of immune system cells and responses that we see occurring in this layer. The papillary dermis contains these little peg-like projections, or these little finger-like projections that are called dermal papillae, and that's what this layer is named for. It's named for these little peg-like projections that kind of interdigitate with or interconnect with the epidermis, kind of like puzzle pieces. So these dermal papillae bind the dermis to the epidermis because the peg-like projections give the papillary dermis a large surface area, so that kind of enhances the ability of the dermis to connect to the epidermis due to its large surface area. We also see that many dermal papillae contain capillary loops that not only nourish the papillary dermis, but also nourish the epidermis, as well as nutrients diffuse out of those blood vessels there. The dermal papillae also house free nerve endings, which function in things like pain reception, and they also contain touch receptors called tactile corpuscles, which we'll see here in a few minutes. These dermal papillae form small ridges, which also tends to shape and contour the epidermis as well. And so if you look very closely on your skin, that is somewhere on your skin other than like the palms of your hand or your fingertips, you see that these ridges create small triangles that kind of section or divide the skin. And what you're seeing is that these are the bumps and the projections formed by the papillary dermis, and the epidermis kind of contours to follow it there. In thick skin, on the other hand, such as the palms of the hand and the soles of the feet, the dermal papillae lie on top of these larger mounds that are called dermal ridges. The dermal ridges kind of in turn cause the overlying epidermis to form epidermal ridges, because the epidermis kind of closely follows the little hills and valleys that are made by the dermal ridges. And so collectively, these skin ridges, so both the dermal and epidermal ridges, are collectively referred to as friction ridges, particularly on the palms of the hand, the fingertips, and the soles of the feet. And these really kind of serve to enhance our ability to grip certain types of surfaces, but they also enhance our sense of touch, or contribute to our sense of touch by enhancing vibrations that are detected by these structures called lamellar corpuscles that are located deeper in the dermis. We'll talk about those guys in just a second here too. So being an areolar connective tissue, the papillary dermis contains an abundance of both collagen and elastic fibers. So collagen is stained in pink in this particular slice of tissue here, this particular section of tissue here, and the elastic fibers are much thinner and they are stained in purple. And so both of these fibers, the collagen and elastic fibers, the elastic fibers in particular give the dermis the ability to stretch, and the collagen fibers really kind of help make the dermal tissue strong and tough. So this really contributes to the flexibility, the strength, and the toughness of the dermis. So there are, as I mentioned earlier, there are touch and pressure sensitive structures that are located within various levels or regions within the dermis. And this table just kind of highlights both of those structures here. We have the tactile or the Meissner's corpuscles, and at the bottom we have the lamellar or Pecinian corpuscles. The tactile corpuscles are located in the dermal papillae, so they're kind of shaped, essentially kind of like these little finger-like projections that really fit quite nicely in the dermal papillae. And in particular, these are found in the dermal papillae of hairless skin, and so these are particularly abundant in places like the nipples, the external genitalia, the fingertips, the soles of the feet, and the eyelids. And in these regions, they function to sense very light pressures on the skin, but also vibration as well. So you may be thinking too that, recall in the epidermis, we saw a structure called the tactile cell. Those are different structures, not only because they are located in the epidermis and tactile corpuscles. So tactile cells are located in the epidermis. Tactile corpuscles are located in the dermis. Even though their names sound pretty similar, they have two different locations and two slightly different functions as well. So recall that the tactile cells sensed very light touch, whereas the tactile corpuscles that we're seeing here in the dermis tend to sense very light pressure rather than touch, and also vibration at low frequency. So tactile corpuscles, therefore, are different in their location and what they sense as well. In the deeper parts of the dermis, we see structures called lamellar or Pecinian corpuscles. Because they're deeper in the dermis, they respond not to light pressure, but to deep, heavy pressures on the skin, as well as stretch and high-frequency vibration. So these corpuscles tend to resemble kind of a cut onion in cross-section, where you've got these multiple kind of layers of overlapping connective tissue that are all surrounding a sensory neuron here. And these are generally located deeper within the dermis, so generally in the reticular dermis, that's much deeper down, and even in part of the hypodermis as well. But recall that the hypodermis is not part of the integumentary system as a whole. So here we see a diagram on the left and a light microscope image on the right of a tactile corpuscle. So as I mentioned, they tend to inhabit the dermal papilla that are found in the papillary layer of the dermis. And they're actually shaped quite nicely for it, because they're kind of these long, kind of oval-shaped structures that fit very nicely within the finger-like projections of the papillary dermis. And they do not ever project into the epidermis, but they do come in very close proximity to the epidermis. So those are our tactile corpuscles. As I mentioned, we also have a very extensive network of blood vessels located in the papillary dermis. The areolar connective tissue that makes up the papillary dermis has a very extensive network here that's collectively referred to as the subpapillary vascular plexus. This term plexus here, I don't know if we've introduced it yet, but this term plexus basically just means a network. And we'll see in this context that this is a network of blood vessels. In later chapters, we'll see structures that are called plexuses, but they are clusters of nerves and things like that. So we'll see this in the nerve. This term is also used to refer to networks of nerves and neurons within the nervous system. So because this subpapillary vascular plexus is in close proximity to the stratum basale of the epidermis, we tend to see that nutrients and oxygen diffuse from these blood vessels and nourish not only the papillary layer of the dermis, but also the stratum basale and the deeper layers of the epidermis as well. So the subpapillary vascular plexus nourishes both the dermis and the epidermis, or at least it nourishes the superficial layers of the dermis and the deep layers of the epidermis. So our second layer of the dermis is the deep reticular layer of the dermis, and this is kind of accounts for the majority or the vast majority of dermal tissue, as the reticular dermis is about 80% of dermal tissue, whereas the papillary dermis is only about 20% of dermal tissue. We have a tissue type change here. We see that the reticular dermis is not made out of areolar connective tissue like the papillary dermis is. Instead, it's made of dense, irregular connective tissue, which, as you recall from Chapter 4, contains mostly collagen fibers. And so these are the fibers that are tough, they resist stretch, and they also resist tearing as well, which this layer really contributes to enhancing the strength of the dermis as a whole. Granted, I will give you that the name of the reticular dermis is a little bit confusing, since the reticular dermis, your first thought may be that it's made out of reticular connective tissue, but it is not. It is made out of dense, irregular connective tissue. There are some reticular fibers in here, but they are not the vast majority of the reticular dermal tissue. The reticular dermis also contains a network of blood vessels that's referred to as the dermal vascular plexus. So this is a plexus that's much deeper in comparison to the subpapillary vascular plexus. And you can see that this dermal vascular plexus has blood vessels that go up to feed the subpapillary vascular plexus. And so the dermal vascular plexus is really what's delivering blood to that subpapillary plexus that we saw a few minutes ago. So the dermal vascular plexus, because of its depth, serves to, or functions to, nourish the deeper dermis and the tissues and structures within the deeper parts of the dermal tissue. So in this layer too, in this layer and in the dermis as a whole, I should say, we have substances that contribute kind of secondarily to the pigmenting of our skin. And so we saw in the epidermis, we saw melanin, which is the brownish black or sometimes reddish yellow pigment that is associated with giving our skin and our hair and our body hair its colors. But it turns out that melanin is not the only substance that contributes to the color of our skin. And in the dermis, the blood that's in our dermal tissue actually helps to contribute to the color of our skin as well. And not just specifically the blood, but not just the blood in general, but more specifically the red pigment hemoglobin that is contained within our red blood cells is what gives our skin, especially fair skin, we tend to get a pinkish hue with people who have fair or lighter skin tones, and this reflects the reddish kind of crimson color of this oxygenated pigment called hemoglobin that we find in our red blood cells. Of course, we have an abundance of capillaries and small blood vessels within the dermis, and so we do have a lot of blood flow within the dermal tissue, and so the hemoglobin that's within those red blood cells contributes to our skin pigment as well. And because light-skinned people or fair-skinned people tend to only have small amounts of melanin in their skin, this makes the epidermis kind of almost transparent and allows the hemoglobin in the blood of the dermis to kind of better show its color through the more fair or lighter skinned individuals. So hemoglobin not only contributes to our skin color when it's well oxygenated, it also contributes to our skin color when it's poorly oxygenated. So when hemoglobin is really poorly oxygenated, the skin kind of tends to take on this kind of blue or grayish tint, which you can see here in this individual's fingertips here. And this bluish-gray tinting is referred to as cyanosis. And this is particularly abundant in things like the fingertips, the lips, the mucous membranes, nail beds, things like that. And so because these structures tend to have a lot of blood flow, and that blood flow is very close to the surface of the skin, it's easy to see when hemoglobin is not particularly well oxygenated. And so cyanosis can be a sign of respiratory or cardiovascular problems. And so just by looking at the skin, just by looking at certain parts of the skin, we can tell what's going on with the blood, with the hemoglobin in that particular individual, and we can use that as a factor in diagnosing respiratory or cardiovascular problems.